Well, we hit 50, so we'll get going. Good morning, everybody. And it's my great pleasure to welcome you to our CART Distinguished Lecture Series. And, in terms of housekeeping, Monica wanted me to remind everybody that you can ask questions. There's a question box. And at the end, in the Q&A, I'll do my best to get through those and ask your questions to Mustafa. It really is a great pleasure. It's a bittersweet pleasure because I love to see Mustafa in person, but having him this way is definitely a treat for us. In any way that he will come west to California, we will always have open arms for Mustafa. He's a professor of neurology at Harvard Medical School and the director of the Rosamund Stone Zander Translational Neuroscience Center at Boston Children's Hospital, where he's developing really innovative translational approaches to neurodevelopmental diseases. And he's truly unusual in that he is a practicing expert pediatric neurologist, as well as a exceptional developmental neurobiologist. And I know his training is exceptional because we did our PhDs in the same lab of Susan Hockfield, and we were in medical school several years apart, but getting Mustafa to join Sue's lab was always considered a great coup because he was always has been one of the smartest people around. We're also going to be recording this talk, I just want to say, just FYI. So I'm going to quickly, introduce, I think he was an undergraduate at Brown University, if I remember correctly, and then did his MD, PhD at Yale. He did his internship and residency at CHOP at the Children's Hospital, and then as well as at Boston Children's Hospital from 1997 to 2000. So he really has been at Harvard really for the most of his practicing life. His work kind of couples really elegant developmental neurobiology, especially in animal systems, and now I think in kind of cellular iPSC-derived systems, but mostly animal models, and very elegant developmental neurobiology with kind of clinical insight. And he's really considered, a leader in pediatric neurology and really one of the greats of his generation in this area. So it's really great to have him here.
Thank you, Dan. That was very kind of you. I have to say it's a huge honor and a great pleasure to present to your group. Of the places I've collaborated with over the years, I think UCLA has been one of the most consistent, and I hope this talk will lead to even further collaborations between our group and the center you have there. So I'd like to start with my disclosures. Due to the translational nature of our work, I work with a number of pharma and biotech companies. In the very beginning, I want to give you my perspective of where I think we stand in autism spectrum disorder today. And the huge success in the field, obviously, over the last 15 or so years has been the identification of these genes that are now associated with susceptibility for developing autism. This, unfortunately, in my mind, as a practicing child neurologist, is a potential handicap in terms of developing treatments, this whole heterogeneity, genetic heterogeneity of autism spectrum disorder. We can think of a few different scenarios where we can develop a broad spectrum treatment that will work for all genetic causes of autism. We think this might not be very practical because we know many of the causes, for instance, in terms of gene dosage, may actually be happening in opposite directions. So having one treatment that will work for all causes of autism might be quite unrealistic. On the other side, we may have to think about a situation where we have to develop different treatments for every gene or every variant, which is an incredibly resource-intensive scenario. So many of us in the field have been hoping for a scenario where there might be convergence either at the cellular or at the circuit level, where subcategories of autism may have some overlap. And we proposed over the years that the mTOR activation, either too much activation of mTOR or too little activation of mTOR, might be a convergence point in the landscape of autism spectrum disorder. So I'd like to tell you the work we've been doing on tuberous sclerosis, a disease in which there's too much mTOR activation as a starting point to understand some of the abnormalities that might be contributing to autism spectrum disorder. So why tuberous sclerosis? First of all, 50% of the patients with tuberous sclerosis are affected with autism spectrum disorder. Very importantly, many of the TSC patients will be diagnosed before birth or at the time of birth because of the cardiac tumors that they develop, and those are easy to diagnose in utero. Cellular mechanisms aberrant in TSC, in particular the mTOR pathway, are being studied from not just from a perspective of neuroscience, but also oncology, transplant medicine, immunology, etc. And very luckily, we have FDA-approved specific inhibitors of the mTOR pathway that can be repurposed to ask specific hypotheses, to interrogate specific hypotheses in this field, both in the preclinical setting and in the clinical setting. Just a very short overview of what the disease is. Tuberous sclerosis is a genetic disease that causes benign tumors in the brain, eye, skin, kidneys, and the heart. It usually presents with seizures. 90% of the patients will have seizures sometime in their life, but half of them will be affected with intellectual disability and or autism. The incidence is roughly 1 in 6,000 to 1 in 10,000 newborns. So we anticipate 50,000 people in the United States, about 1 million people worldwide to be affected with this disorder. And the genes TSC1 and TSC2, in which one mutant allele is sufficient to have the diagnosis, have been known for about 30 years now almost, and have been studied in various settings, including, aside from neuroscience, in oncology very intensively. It turns out that TSC is a disease of cell size dysregulation. Here on the left are images of a brain specimen taken at the time of epilepsy surgery. You see the presence of these giant cells, which are pathognomonic for this disease. But any organism, any cell that's missing the TSC1 or TSC2 genes shows cell enlargement. So here's a typical Drosophila eye. Here's a TSC1 mutant Drosophila eye. The whole eye is bigger. Ommatidia that make up the eye are bigger, and the hair cells on the Drosophila are bigger if they're missing the TSC1 gene. The pathway that results in this phenotype is summarized here. There's a GTPase called Reb, which activates a kinase called mTOR, which stands for mechanistic target of rapamycin. And together, when they're active, that results in increased protein synthesis and cell growth. TSC1 and TSC2 proteins form a protein complex that puts a brake on Reb. In fact, TSC2 is a gap GTPase activating protein for Reb. So their normal function is to put a brake on protein synthesis and cell growth. And as you can imagine, if you're missing either one of these proteins, you have overactivation of the mTOR pathway and too much cell growth. Cells have taken advantage of this pathway in various ways. So growth factors such as insulin or BDNF activate a kinase called AKT that phosphorylates and inhibits this complex, resulting in cell growth. And on the other side, if you break down your ATP to AMP, you have activation of this kinase called AMP kinase, which phosphorylates TSC2 on a different set of residues and activates this brake, putting a brake on protein synthesis, so a starving cell doesn't continue to grow. But the most important aspect of this connection between TSC and mTOR is the fact that we have natural and very specific inhibitors of this kinase, mTOR, and they've been known for several decades now. Rapamycin, which was the original Rapalog, was identified on Easter Islands in the Pacific Ocean. Its chemical structure is shown here. And both rapamycin and its analogs that have been developed by pharmaceutical industry have now been in clinical trials. In TSC, the first approval of the Rapalogs was for the brain was everolimus, which is a 1-methyl difference from the original rapamycin molecule. And everolimus was shown to be effective for supramandibular giant cell astrocytoma, which is a rare brain tumor that's seen in tuberous sclerosis patients. This required only 28 patients because the effect size was so large that the FDA approved this drug after a small size study back in 2010. The second indication for brain manifestations was intractable seizures. Everolimus was tried in a large phase 3 trial of around 300 patients and was shown to be effective, leading to this publication in 2016 and finally its FDA approval around 2018. What we've been particularly interested in the lab is the relationship between tuberous sclerosis and autism. And Shefali Jesti, when she was a child neurology fellow here, wanted to ask the question, what is the true incidence of autism in the TSC cohort? And there were, in the literature, in retrospective studies, the numbers range from 17% to 80%. So she actually collaborated with Patrick Bolton's group in the United Kingdom and looked at a cohort of prospectively studied TSC patients from 2 years of age to 5 years of age. And as you can see here, roughly 50% of the patients with TSC will have either full-blown autism or autism spectrum disorder diagnosis, and the other 50% will not. One of the most common questions I get asked is whether the symptomology of autism in TSC is different from non-syndromic autism. And there are several lines of evidence to suggest that it is not. Shefali did this study when she was at UCLA, looking at the ADAS subcategories of scores of TSC patients with autism shown in purple here, and non-syndromic autism shown in orange. As you can see, there's a significant overlap between the symptom profiles of TSC patients with autism and non-syndromic autism. So what causes autism in TSC? We don't currently have the full answer. The original studies suggested that having bilateral temporal lobe tubers were necessary, but not sufficient for having tuberous sclerosis. This was a 1.5 Tesla MRI study done by Patrick Bolton and colleagues. Unfortunately, several other studies have failed to show the same correlation. So the relationship between tuber load or location and autism is still up for grabs in terms of needing further analysis. We have really focused on the non-tuber pathology of TSC and have been pursuing the hypothesis that TSC1, TSC2 regulate neuronal connectivity and function through the mTOR pathway. And over the years, we have followed various different neuronal roles for TSC1, TSC2 proteins in the brain. We first looked at the role of TSC proteins in axon guidance and specification. Then we turned to their role in mitochondrial dynamics in axons. We looked at non-cell autonomous effects of TSC loss on myelination through regulation of oligodendrocytes. We've also focused on epilepsy, cerebellar circuitry, and through the work of Jonathan Lipton, we've focused on the role of TSC proteins in circadian rhythms, which is a major manifestation of tuberous sclerosis in terms of abnormalities in sleep patterns and circadian rhythm. What I'd like to do today is to focus just on our work on epilepsy and cerebellar circuitry as it relates to manifestations that we see in the clinic that are extremely common in our patients. So first, the work on epilepsy. Essentially, every mouse model of tuberous sclerosis that has been developed in various labs, including our own, results in seizures. And these are spontaneous seizures for the most part. Here's a subset of the studies that have been done to date. And regardless of whether the inactivation is in an astrocytes or neurons, inhibitor neurons, or excitatory neurons, the result in phenotype is seizures. There's one important distinction, which is a loss of TSC genes in the cerebellum, which I'll come back to later in the talk. We first focused on a synapsin-Cre TSC1 mutant developed by David Kwiatkowski and worked with him to further characterize this mutant almost 15 years ago now. And what we found in that mouse model is that null mutations in TSC1, just in the neurons, postmagnetic neurons driven by the synapsin-Cre promoter, results in a mouse model that has early onset epilepsy with seizures starting around 3 weeks of life, which results in mortality of all the animals by about 60 days of life. We then tested whether rapamycin could change the natural history of this disorder in this mouse cohort. And we treated these mice with either rapamycin or everolimus. At the time, it was called RAD-001. We first looked at the pharmacokinetics of both of these compounds and chose appropriate dosing to get enough of the compounds into the brain. And what we saw was that the vehicle-treated mice died from seizures by 60 days, as you can see here. But rapamycin-treated mice did not show any seizures, and 90% of them survived as long as you kept them on the medication. If you stop the medication by day 30, they started having seizures about a month later and started showing early mortality. And we saw almost identical results with everolimus treatment. So in mouse models, these compounds, rapalogs, are extremely effective in essentially rescuing full range of the cohort in terms of stopping seizures and increasing the lifespan. In the human clinical trials, the picture was a bit more complicated. This is a study that I mentioned earlier that was published in Lancet by Jackie French and colleagues. And here's one panel from that paper. As you can see, there were 3 cohorts: placebo-controlled treated patients, everolimus-treated patients at a low dose, and everolimus-treated patients at a high dose. And roughly 15% of the placebo group showed a positive response, which is defined as greater than 50% reduction from baseline frequency. In the low-dose everolimus group, the response rate was 30%, and in the high-dose everolimus group, the response rate was about 40%. And the other 60% of the patients did not respond, even with just a 50% reduction from baseline. And remember, this response rate does not mean the patients become seizure-free. They just go from 10 seizures to 4 seizures, let's say, per day when they're treated with this medication. So in terms of unmet need in intractable epilepsy in TSC, we have a long way to go. So we've been trying to come up with alternative approaches to treating epilepsy in tuberous sclerosis patients. Our initial mouse model, the synapsin-Cree TSC1 mouse model that I mentioned a few minutes ago, it was a very fragile phenotype, in the sense that if you try to do EEG recording in this mouse model, they would not survive the anesthesia required to place the electrodes for continuous recording. So we wanted to develop a more translational relevant mouse model that had spontaneous seizures that were 100% penetrant, but was able to survive electrode placement, so we could do long-term EEG monitoring over days and weeks, just like we do in some of our patients in the EMU. And we wanted to test whether we could modulate this phenotype using small molecules. We were lucky that David Kwiatkowski developed a hypomorphic allele of TSC2 that removes the exon 3 near the end terminal of TSC2, but roughly 8% of functional TSC2 protein expression is preserved in this mouse model, and that seems to be sufficient for this mouse to be relatively healthy, but still shows symptoms of hyperactivity and seizures in all of the animals. So we, in our IDDRC core at Boston Children's, we have this epilepsy monitoring unit for mice. We have 16 cages that are transparent with night vision cameras where we can watch the clinical seizures, and we have a EEG receiving plate underneath the cage that receives continuous EEG from this mouse. And we can record them either continuously for 2 weeks or intermittently, so we can look at the phenotype in response to treatment. The first question we wanted to ask was raised by a very important paper in the field that came out around 2011 by Mark Baer's group at MIT. We had been collaborating with them, and we were really quite intrigued by their results shown in this paper. What they showed was that Fragile X and TSC have opposite phenotypes in terms of hippocampal LTD, such that Fragile X knockout mice have increased LTD, whereas TSC2 knockout mice have decreased LTD. And if you cross these mice such that you have a double heterozygous, those mice have normal LTD, just like the wild type litter mates. So this was suggestive of the fact that you could treat the Fragile X mice with a mGluR5 negative allosteric modulator, whereas you would have to treat the TSC mice with an mGluR5 positive allosteric modulator to get back to the physiological state. These results were really impressive, and they were done in the TSC2 heterozygous mouse model, which is a mouse model that doesn't have any seizures. So the first question we wanted to ask is, what happens if we use this approach of treating the animals with TSC with an mGluR5 PAM but used our mouse model that actually has spontaneous seizures at baseline. So we did this study where we took the hypomorphic allele of TSC2 and looked at the seizure and behavioral phenotype of the TSC2 hypomorphic allele in response to mGlu5 modulation. We collaborated with our colleagues at Roche. They gave us a compound that was a positive allosteric modulator of the metabotropic glutamate receptor type 5. And we first looked at the open field behavioral analysis of this mouse. At baseline, our knockout mice are significantly hyperactive, as you can see here, compared to control litter mates. Upon PAM treatment, they become even more hyperactive, and this also happens in the control case. But what was really worrisome to us was the fact that upon PAM treatment, even a single acute administration of PAM with one dose, these mice showed a significant increase in seizure frequency, and many of them died after a single dose. So these results were quite surprising to us and concerning in terms of the efficacy and, of course, in safety of using mGlu5 positive allosteric modulators in TSC. So we decided to test the opposite pharmacology. Instead of a positive allosteric modulator, we wanted to test what happens with a negative allosteric modulator. In this case, we used CTEP, looked at the open field assay for the two genotypes. Our mutant mice are hyperactive at baseline, but upon NAM treatment, they become less hyperactive. But, and equally importantly, the seizure frequency dropped upon chronic NAM treatment. So these results suggest that at least in this mouse model, mGlu5 negative allosteric modulators could potentially be helpful in terms of improving the behavioral and seizure phenotype. So a Swiss company called Noema is actually about to start a clinical trial with an mGlu5 NAM in the United States. It's already started in Europe, and we're going to be one of the sites testing this mGlu5 NAM based on the preclinical data that I showed you. So we're quite excited about the possibility of using mGlu5 negative allosteric modulators as adjunctive therapy in TSC and intractable epilepsy. We also, in addition to our mouse models, as Dan mentioned, we are interested in using cell-based assays to identify novel compounds for treating epilepsy. In our IDDRC core, we have a human neuron core that got some funding from Massachusetts Life Sciences. So we're able to populate it with 5 PhD level scientists and 5 research assistants. This has the capability for making pluripotent stem cells and turning them into different type of neurons from either patients at Boston Children's or other centers. And the important aspect is that we're aiming to have standard operating procedures for different cell types, such as excitatory, inhibitory, dopaminergic, spinal motor, and sensory neurons, so we can reproducibly make these subsets of neurons. And we have some small molecule libraries and equipment or high content imaging equipment that will allow us to do screening. So I'll talk a little bit about the work that we've started to do applying these technologies to tuberous sclerosis. This work is actually led by a UCLA MD PhD who's now on faculty here at Boston Children's. Kellen Winden came from Dan's lab. He has been working with 3 genotypes of iPSCs, wild type iPSCs, patient-derived heterozygous iPSCs, and a null line in which we induced a second allele mutation in the patient-derived iPSCs. So comparing these 3 genotypes, as you can see, the TSC2 null neurons are significantly larger compared to the other 2 phenotypes. Kellen first looked at the expression of TSC2 protein. As you can expect, as seen on this Western blot, there's a gene dosage dependent reduction in TSC2 protein expression. At the same time, we see an increase in mTOR activity as measured by phosphorylation of S6 ribosomal protein in a, again, gene dosage dependent way. And using this allelic series, Kellen started looking at the morphology and function of these neurons. The heterozygous cells are larger in cell body size compared to wild types, and the null cells are even larger. And the neurite length is also increased. We had seen these phenotypes in mouse and rat neurons in the past, but this confirms that the same phenotypes are seen in human neurons. But more importantly, we are starting to see differences between wild type and heterozygous cells that we had not appreciated in rodent cultures before. And then Kellen started looking at the function of these neurons in culture. So here's using multi-electrode array recording. Each row is represents an electrode in these multi-electrode setups. He's comparing wild type, heterozygous, and null cells again. And as you can see, wild type cells fire at some frequency. There are certain moments where they synchronize and fire at the same time. Heterozygous cells fire at roughly the same frequency, but there might be an increase in the synchronicity. And then finally, if you look at the null cells, as you can see, these cultures are much more hyperactive as network, and there might be increased synchronicity. So Kellen was able to quantify those. The normalized mean firing rate in these MEA cultures is much higher for the null cells compared to the other two genotypes. And then if you look at the synchrony index, heterozygous cells are in the middle, wild type cells are at the lowest synchrony, and then our null cultures at the highest synchrony. So this gives us a cell-based platform where we can start testing compounds, and we obviously tested as a proof of concept first rapamycin treatment. And you can see here, this is the DMSO or vehicle-treated null cultures. If you treat them with rapamycin for over 20 days, the culture shows much less firing frequency, roughly the same firing frequency as the wild type cultures. Importantly, if you wash out rapamycin, the firing frequency comes back up, highlighting the fact that the treatment has to be maintained. Now we have a platform that we can then start to use to do high content imaging studies. And we started using this platform first on rat neurons, and we hope to expand it to human neurons as well. Our first paper came out a couple of years ago, where Alessia Leonardo in the lab took the lead on a phenotypic screen using TSC deficient neurons and did a drug screen looking at mTOR activity and cilia phenotype. It turns out that TSC loss results in abnormalities in cilia, in primary cilia in neurons. Using this platform, Alessia was able to identify heat shock machinery as a potential druggable target, and it's an area that we're working actively on to see if those compounds could be developed into safe potential treatment targets in neurological disorders. I should say the work we've been doing with the human neuron core has led to several important areas of learning. One important area that we started appreciating early on is how difficult it is sometimes to reproduce the results in culture using these relatively fragile and sophisticated human neuron cultures. So collaborating with the stem cell groups in the IDDRC network, we put out this paper last year where Nikesh Anderson in lab took the lead in doing an analysis of recent stem cell derived neuron papers in intellectual and developmental disorders. This was a collaboration with the whole cross IDDRC human stem cell working group, and really led by Anita Bhattacharya at Wisconsin and Kristen Kroll at University of Washington, St. Louis. I'm sorry, Washington University in St. Louis. And what she showed in this paper is that actually the quality, the QC score in terms of what the papers were reporting was going down over the years. This is a timeline from 2010 to 2018. Even though these publications are reporting larger and larger cell line numbers, the quality controls that were reported, either performed or reported in the papers were going down. So we actually have some specific recommendations about how to perform these studies as well as how to report these studies that could potentially increase the reproducibility and make them more impactful for drug discovery and impact on the quality of life of our patients going forward. I want to move from epilepsy to our work on autism using cerebellum as a starting point. This work was started by Peter Tsai, a child neurologist in the lab, who now has his own lab at UT Southwestern, and has been since continued by Maria Sandberg, a very talented postdoc in the lab. I just want to sort of put this timeline here because as Dan mentioned, we were in the same lab, and in Sue Hockfield's lab, my rotation project was to stain some cat cerebellum sections with an antibody that Susan had developed. And that antibody identified a specific cell type called the Lugaro cell in the cerebellum. And that was one of my first papers. Back in 2012, we worked on the mouse cerebellum with Peter Sahin, and more recently, we've been working on the human cerebellar cultures. And I'll tell you a little bit about that story. So, as child neurologists, obviously, we think of the cerebellum more from a motor function, but obviously, cerebellum has many non-motor functions. This has been shown by the work of Jeremy Schwammen and many others in the field now, that it is involved in executive function, language, attention, working memory, and various types of behavioral manifestations. In autism spectrum disorder, one of the more common findings in neuroimaging studies is a decreased vermis volume and abnormal white matter in the cerebellum. In pathological series, which are obviously rare in the autism field, the most consistent finding in the literature is a reduction in cerebellar Purkinje cell numbers. And then finally, Kathleen Propoulos, when she was here at Boston Children's, did a study where she followed newborns with isolated cerebellar hemorrhage. And when she did assessment for autism later in life, what she found was that 37% of those with isolated cerebellar hemorrhage in the newborn period would go on to develop autism spectrum disorder. So Sam Wang, one of my collaborators at Princeton, published this chart where he's mapped out different risk factors for autism. And having an autistic identical twin obviously increases your risk significantly, but the second highest risk factor is having cerebellar injury at birth, highlighting the importance of cerebellar circuitry in autism. In TSC, there's been a number of studies suggesting that cerebellar abnormalities on MRI can be seen in about 30% of the patients, even though by bedside testing, you don't detect any dysmetria or ataxia. Purkinje cell loss has been detected in autopsy specimens in TSC patients. There are two cohort studies where cerebellar pathology, in particular cerebellar tumors, have been correlated with autism diagnosis in individuals with tuberous sclerosis. And then finally, a very interesting study by Harry Shigani showed that if you do PET scanning, 2DG PET scanning in patients with TSC, those with autism have hypermetabolism in the deep cerebellar nuclei compared to those with hot autism. As you know, cerebellar cortex inhibits the deep cerebellar nuclei, so hypermetabolism in the deep cerebellar nuclei implicates a hypofunction of the cerebellar cortex. So Peter Sahin decided to test this hypothesis that cerebellar dysfunction contributes to the pathogenesis of ASD and TSC. And he developed a mouse model in which he deleted TSC1 gene under the L7-CRE promoter. These L7-CRE is only expressed in the Purkinje cells in the brain, and it causes recombination by postnatal age 6. These animals turn out to have a normal lifespan. We looked at 3 genotypes, control litter mates, heterozygous litter mates, and then null mutants, and they all gain weight and are relatively healthy. So we can do behavioral testing on this mice colony. Peter first looked at the Purkinje cell staining in the cerebellum. Heterozygous mice have equal number of Purkinje cells as the controls, and there's no enlargement of cell size, as you can see here. However, the null mutants, the cells get much larger, as you would expect from mTOR activation, but they also seem to die off, and we have evidence that this happens due to oxidative stress, such that by about 4 months, you only have about 25% of the Purkinje cells left. This is quite reminiscent of what's been published in the literature from human patients with TSC. There's a significant reduction in Purkinje cell density in the cerebellum of patients with TSC compared to non-TSC controls. What Peter did was then to collaborate with Jackie Crawley's group and do behavioral assessments in this mouse. We used a 3-chamber approach that Jackie developed, and our control animals interacted with the novel animal much more than the novel object, as you would expect. Time investigated is shown at the bottom. However, our het and null mutants did not show that preference. They essentially spend equal amount of time with the animal as the object, and time investigating again is shown here. So missing either one or both copies of the TSC1 gene just in the Purkinje cells in the cerebellum seems to be sufficient for giving the social interaction phenotype. As a second behavioral axis of autism-like behavior in the mouse model, we turn to cognitive inflexibility, and we use this reversal learning paradigm using the water T maze. You teach the location of the platform on one side of the T maze arm, and then the animals learn that over 3 days. Our mutants have no deficit in that hippocampal-based task. But then if you move the platform to the opposite arm of the T maze, the animal has to forget the original location and learn the new location. And our null mutants especially show deficits in that reversal learning paradigm. To look at a bit more objective physiological assessment of the neuronal function, we turn to Wade Regier, one of my colleagues at Harvard Medical School. He recorded from cerebellar slices and showed that there was a gene dosage dependent reduction in excitability of Purkinje cells, such that the heads were less excitable and fired less frequently than controls, and the null mutants fired even less frequently, as shown here. And then finally, Peter in this paper asked the question whether rapamycin could rescue the phenotype of this mouse model. And he treated the mutants and the controls with rapamycin or vehicle. The vehicle treated mice showed a reduction in Purkinje cell numbers, which was completely abrogated by rapamycin treatment. But more importantly, there was a functional improvement. So in the social approach, 3-chamber social approach test, vehicle-treated mice spent equal amount of time with the animal as the object, but rapamycin-treated mice spent much more time with interacting with the animal than the object, just like their litter mates, control litter mates treated with rapamycin. So we were really encouraged by these results, but as a neurologist, we always wonder whether we could translate these results into the clinical setting. And what was troubling us about these findings was that we had started the treatment at P7, roughly the same time that the Cre turned on in this mouse model. Essentially, as we were deleting the TSC1 gene, we were starting to treat with the mTOR inhibitor. To be more translation relevant, we wanted to ask the question, if we wait up for a while, in terms of the development of the symptoms in this mouse model, can we start the treatment later in life and still rescue the behavioral deficits? So what Peter did was to compare starting treatment at 6 weeks versus 1 week, and did the same assessments. At 1 week, as I showed you earlier, we can rescue with rapamycin treatment, starting the treatment at 1 week, we can rescue the 3-chamber social approach phenotype. If you wait for 6 weeks and then treat for 4 weeks after that, you can still rescue the social approach phenotype, as you can see here. Or when we turn to the reversal learning paradigm with the T maze, water T maze, what we were surprised was that even though we could rescue that water T maze assessment if we do the treatment at 1 week, we could no longer rescue it if we started the treatment at 6 weeks. These mice also have increased grooming, significantly self-groom, and this was also not rescued by rapamycin treatment starting at 6 weeks, suggesting that the two significant deficits in this mouse model, the social approach and the repetitive behaviors, may have different windows of treatment. Peter then asked the question, is the window for social approach deficit infinite for treatment, or does it close at some point? Yeah, the answer appears to be the latter. If you start treatment at 10 weeks and treat for 4 more weeks, you can no longer rescue the social approach phenotype, suggesting that the window for social approach is larger in terms of rapamycin treatment, but it's not infinite. So this is just a summary of the different experiments I described. If you do the treatment starting at 1 week, you can rescue essentially all of these phenotypes. If you start the treatment at 6 weeks, you can rescue the social approach phenotype, but you cannot rescue the grooming or the water T maze deficits. And if you start the treatment at 10 weeks, you cannot rescue either one of those deficits. Why is this important? One, from a fundamental basic science perspective, is interesting because this suggests that cerebellar-based deficits in this mouse model in terms of social approach versus repetitive behaviors may have different windows of treatment and potentially different circuits underlying them. And that's something that Peter is following up in his lab. The other aspect is it raises questions about the onset of treatment initiation for clinical trials. We had actually done a clinical trial using everolimus in our patients with fetal tuberous sclerosis. And in this study, we did a treatment study between 6 and 21 years of age. And it was a placebo-controlled trial. And we did neurocognitive assessment at 0, 3, and 6 months. And we published the paper back in 2017. And we failed to show an improvement in the group treated with everolimus compared to those treated with placebo. And one possibility is that by the time you have these deficits, neurocognitive deficits, for 10, 15, 20 years, it might be too late to do a 6-month treatment with an mTOR inhibitor, improve those deficits. So we're, as I'll tell you in a few minutes, we are planning trials and executing trials by studying treatment much earlier. I'm going to turn to our work on human Purkinje cells. One of our insights from the mouse model was that TSC deficient Purkinje cells have abnormalities both morphologically and functionally. We were wondering whether we could see the same phenomena in human Purkinje cells. So we collaborated with Mary Beth Hatton at Rockefeller, and with their help developed this differentiation protocol to make human Purkinje cells from iPSCs. Maria Sundberg developed this protocol, and by the time you do purification, you can get these cultures to be roughly 80% positive for Purkinje cell markers such as PCP2. Importantly, we saw the same physiological deficit that we had seen in the mouse slices. So our heterozygous Purkinje cells, shown in red here, are less excitable. They fire less frequently than wild-type Purkinje cells. An important rapamycin treatment, you can rescue this deficit in terms of lowered firing rate back to wild-type levels. So this gives us a functional platform to test molecules in cell culture and looking at this excitability phenotype. We're quite excited about that possibility of using this as a translational assay. At the same time, one of the things that Maria noticed in her gene expression profiles was that there were modules that showed reduced gene expression in TSC deficient Purkinje cells. And those modules were really enriched for FMRP targets, Fragile X mental retardation protein targets. There was also some reduction in FMRP levels in iPSC-derived Purkinje cell cultures. So that led us to ask the question, is FMRP and TSC somehow interacting in Purkinje cells? And what is the functional significance of that interaction? So Jasbir Dalala and Kellen Wind in the lab have been pursuing this interaction. First, what they did was to purify Purkinje cells from GFP tagged animals with and without TSC1 expression. And when they looked at the overall gene expression, what they saw was that loss of TSC1 in Purkinje cells results in an overall downregulation of gene expression in a larger number of genes compared to genes that are upregulated, as you can see on this volcano plot. Within the genes that were downregulated, however, there was a significant enrichment of FMRP targets again. This led us to ask what happens at the ribosomal interaction of the mRNAs. So we did trap using a ribosome-tagged mouse model. We collaborated with Miriam Hyman's lab. We pulled out ribosomes, GFP-tagged ribosomes, and looked at the mRNAs bound to the labeled ribosomes. And when you look at the volcano plot here, they're essentially balanced, both in upregulation and downregulated genes, as it's shown on the heat map here too. So trap or ribosome bound mRNAs do not show that reduction in transcript levels that we see in the whole transcriptome. This is another way of looking at the data. If you look at the transcriptome versus the ribosome-associated mRNAs, there are mutants show a reduction in the transcriptome that is not seen in the ribosome association, suggesting that there's a compensatory increase in relative ribosome binding or translational efficiency of FMRP targets back to control levels. So one wonders if there is a reduction in transcriptome or gene expression, and there's a compensatory increase in ribosome binding. What is this final effect on the protein levels? We started looking at them, some of the targets, and one of the targets is SHANK2. SHANK2 is an FMRP target. It turns out that by protein labeling, there's a reduction in the mutants compared to the wild types. So I think it might be that even though there's a compensatory increase at the translational efficiency, that the final effect might depend on the overall expression at the transcriptome level, at least for a subset of proteins such as SHANK2. We're very interested in understanding this complex relationship between FMRP targets and TSC, at least in Purkinje cells, but also potentially other cell types in the brain. In the last few minutes, I want to turn to our clinical work. TSC is unique among the autism spectrum disorders in the sense that you can identify these patients very early in life because of the presence of these cardiac rhabdomyomas. Among fetus or newborns with multiple cardiac tumors, the chance of having TSC is roughly 95%. So given the fact that half of these patients will go on to develop autism, we've been trying to ask a simple question: can we detect which infants born with TSC will go on to develop autism? And we've been collaborating with Simon Warfield, Chuck Nelson, and Stefan Jeste on that question for many years now. We've been doing neurocognitive assessment, diffusion imaging, as well as neurophysiological assessment of these babies. The first study we did at a single site in Boston was led by Julian Peters, where he looked at the diffusion tensor imaging using corpus callosum as a region of interest. And he compared controls without TSC, TSC patients that are not on the spectrum, and then finally TSC patients with autism. And as you can see, these two groups look very similar to each other visually, but also by fractional anisotropy. They're almost identical. However, in TSC patients with autism spectrum disorder, there is significant microstructural abnormalities such that fractional anisotropy of the corpus callosum is significantly decreased. This was a single site study with relatively old patient cohort. So we wanted to collect a large group of patients and using an ACE Autism Center of Excellence network funding, we collaborated with 5 centers, including UCLA, to collect babies born with TSC. And we did prospective assessments with neuropsychological assessments, MRI and EEG. And this is a huge data set that we're still in the midst of analyzing. The first results really focused on EEG as a potential predictor of impending seizures. Joyce Wu led this paper that we published back in 2016. And our main finding was that in many cases, if you do prospective EEG on babies born with TSC, is that first EEG is normal, as shown with these white bars. Then the EEG becomes abnormal with interictal spikes, shown with these gray bars. And then finally, clinical seizures start. So there is a window in which the EEG is abnormal, but the clinical seizures have not started. And if you average this, the epileptiform discharges start around 4 months, whereas the clinical seizures take another 2 to 3 months to start. So we have roughly a 2 to 3 month window where we can intervene. This observation led to the PREVENT trial that's still ongoing at 14 sites in the United States, led by Martina Bebin. PREVENT stands for Preventing Epilepsy Using Vigabatrin in TSC patients. And our primary endpoint is developmental impact of early versus delayed treatment with vigabatrin. So the babies are randomized to either early treatment with vigabatrin when their EEG becomes abnormal, or they get moved to the clinical seizure arm when the clinical seizures start, and they get treated with vigabatrin at the time of clinical seizure onset. And we're looking at the developmental assessments at 24 and 36 months, as well as the severity and onset of epilepsy. Now, this is using vigabatrin, which is a commonly used anti-seizure medication for infantile spasms and tuberous sclerosis. Another follow-up study is about to start. It's called STEPS, TSC STEPS, stopping TSC onset of progression with sirolimus, which is another name for rapamycin. And this is a phase 2 study. It is not funded by the NIH, but it's funded by the FDA. It's going to be at 8 sites around the country. And the babies will be randomized to everolimus or placebo. And we're going to look at, they're going to be treated with sirolimus to see if we can prevent or delay the seizures in infants with TSC. and the onset of treatment will be between 0 and 12 months, is led by Darcy Kruger. So I want to end here. I just talked about tuberous sclerosis 1, tuberous sclerosis 2 genes. We think there's at least another several hundred genes implicated in autism spectrum disorder. How can we start to find convergence among these genes? As a first step, we proposed a comparative analysis of 3 disorders that impinge on the mTOR pathway, obviously tuberous sclerosis, but also P10 hamartoma tumor syndrome, and also SHANK3 mutations leading to Phelan-McDermid syndrome. We have this rare disease grant called Developmental Synaptopathies Consortium. We are doing neurocognitive assessments as well as some biomarker development in these 3 disorders at 11 sites around the country. And we believe this comparative of the biology of these 3 disorders that have a convergence in the same biochemical pathways and cells might give us ideas for potential treatment, and learnings from one disorder may have implications for the other. So for instance, we just finished a clinical trial using everolimus in P10 patients, and we're just about to report on that. I'm going to stop here and highlight many people that contributed to this work, including Kellen Winden, Peter Tsai, and Maria Sandberg that I mentioned, as well as other members of my lab, and many collaborators that are listed here and not listed here, unfortunately, over the years that contributed to this work and our funding sources. And I'd be delighted to answer your questions. Thank you very much for listening.